So both speakers today are from the visual cognition and
neurodynamics of visual cognition.
Neurodynamics of visual cognition group, led by Professor Radoslav Schifre. And we will start with Alessandro Gifford talking about experimentation on in silico neural responses from encoding models. Stage is yours.
So thanks for organizing, Timo. Okay, so in this talk, I will focus on a research paradigm based on experimentation on in silico neural responses generated by encoding models, and argue how this paradigm can benefit neuroscientific research. So now by encoding models, during this talk, I will be referring to algorithms that take a stimulus as input, such as an image, and predict the corresponding neural response. And I will be referring to this predicted neural response from encoding models as in silico neural response, therefore computer generated, which is in opposition of in vivo neural responses, which are neural responses that are recorded from real brains. So I will begin by introducing this paradigm of experimentation on in silico neural responses, and also introduced the neural encoding dataset, which is a resource that we created to facilitate this paradigm. I will then give an example of how we use this paradigm in one of our research projects, where we investigated the representational relationships between visual areas through a new method called relational neural control. And finally, I will address some of the limitations of this paradigm and argue how these limitations can be addressed by, for example, by challenges in neuroscience, such as the Algonaut Project Challenge. So let's begin with the section on experimentation on in silico neural responses by first talking about in vivo large-scale neural datasets. So in vivo large-scale visual neural datasets are datasets that are recorded on actual brains that are larger than usual in the trials that were presented to single subjects or in the amount of conditions that were presented. So they consist of datasets in which single subjects are extensively sampled with visual stimulation. Usually single subjects undergo several sessions. Examples of these datasets are, for example, the natural scenes dataset, which is fMRI responses to over 70,000 naturalistic scenes, or the SeeNeuromod initiative, which consists of a massive collection of fMRI responses to different types of stimuli, from still images to movies, but also to video game playing, or the bold moments, again, fMRI responses to over 1,000 3-second videos, or also data sets coming from the Things initiative, which consists of different modalities, different data sets, fMRI, MEG, or EEG, which all consist of neural responses to the same collection of 26,000 naturalistic images. So during the last few years, there has been a sharp increase in the usage of these data sets during research by different groups. And some of the reasons, some of the advantages is that they are multi-purpose tools, namely that the same data sets can be used to address many different research questions, but also be used for different types of research, from hypothesis testing to exploratory research. They consist of big data, they are large, and therefore they enable training on neural data of machine learning and deep learning models, which are data hungry. They typically consist of naturalistic stimulation, so they promote the investigation of how the brain functions in day-to-day settings. And they're also freely available, so anyone can download them and use them pretty simply. Now, despite their advantages, they also come with limitations. Specifically, they're pretty expensive to create, and they take a really long time to acquire. And once they are acquired, they're also restricted to the stimuli collected during data collection. So now the question is, how can we retain all of the benefits that these large-scale data sets have, while at the same time addressing these limitations? And we can do this with neural responses and silico neural responses coming from encoding models, since these neural responses are cheap and quick to generate, and they're unrestricted to any stimulus. And specifically, we can leverage existing large-scale neural data sets to train encoding models. And we can use these encoding models to generate in silico neural responses to stimuli of any stimuli of your choice, and then perform experiments on these in silico neural responses. And by experiments, I mean the same experiments that we're all used to, from hypothesis testing to method development, exploratory research, results validation, model development, and also neuro AI research. So then the advantage of in silico neural responses from encoding models is that they can easily scale up to neural responses for hundreds of thousands or even millions of stimuli. And this rapid expansion or generation is resource efficient in that it saves enormous amounts of time and money compared to in vivo recording from real brains. And as a consequence, can greatly speed up research. Now, just imagine, instead of taking you weeks to collect some neural responses, you can have an encoding model generating the same amount of data in only a few minutes. And as a consequence, you can test and try out way more things. Now, this does not suggest that traditional data collection efforts, so data collection from in vivo brain, from real brains, should be eliminated. Instead, this framework proposes a virtuous cycle where fast-paced research is done on in silico data generated by encoding models, and then the findings deriving from this in silico research are validated on in vivo data collected from real brains. And then this validation in turn informs new fast-paced in silico experimentation. So then to promote this research framework, we train computer models of neural visual stimulation for large amounts of images and then made them available as a resource to the community. And since we wanted to provide users with both a good spatial and temporal resolution, we trained encoding models of both fMRI and EEG responses to naturalistic images. For fMRI, we trained feature-weighted receptive field encoding models using the natural scenes dataset, fMRI responses of 8 subject to 73,000 natural scenes. And these models had higher prediction accuracies for early visual cortex, where they accounted for up to 65% of the explainable variance in V1, and accounted for almost 40% of the explainable variance along the ventral stream. For EEG, we trained vision transformer-based encoding models on things EEG2, EEG responses of 10 subjects to over 16,000 images from the things database. The prediction accuracy reached the noise ceiling level at 100 milliseconds after stimulus onset and remained close to noise ceiling level for the entire epoch.
Can I ask a question?
Yeah.
Can you say why you use different models for the two?
Okay, so the reason is that in this case, this is a feature with a receptive field, meaning that it takes into account receptive fields and retinotopy, which is specifically a feature of fMRI, but wouldn't be a feature for EEG. So this was the first reason. The second reason is simply that these models perform better on EEG and these perform better on fMRI. And as I wanted to maximize prediction accuracy, this was the choice. And we then made these encoding models available as an open resource called the Neural Encoding Dataset. And also created a GitHub package so that you can use the trained encoding models to generate in silico fMRI and EEG responses to any image of your choice with one line of code. To help you familiarize with the resource, Neural Encoding Dataset also comes with a manual and tutorials. And to facilitate research on in silico data, the Neural Encoding Dataset also comes with pre-generated fMRI and EEG responses to over 150,000 images from the 3 most widely used image sets in neuroscience research. So then, to summarize this first part, I talked about large-scale, in vivo large-scale neural data sets. I talked about their advantages, but also their limitations, meaning that they're expensive to collect, take a long time to acquire, and once acquired, they're restricted to the stimuli that were presented during data collection. And I proposed in silico neural data sets as a way to address these limitations, in that in silico data is cheap and quick to generate and is unrestricted to any stimulus. Yeah, so I was proposing in silico data as a way to address these limitations on in vivo large-scale data sets. I then introduced the paradigm of experimentation on in silico neural responses, talking about the synergy between in silico and in vivo data, and finally introducing the neural encoding data set as a resource available to the community, consisting of trained encoding models and code, so that you can easily generate fMRI and EEG responses to images of your choice, and then use them for experiments. Okay, so now in the second part, I will give you a concrete example of how we used in silico neural responses generated by encoding models in one of our experiments. And specifically, in this experiment, we were interested in the representational relationships between visual areas. And for this, we created a new method called relational neural control. So the topic of this project is representational content. which is visual information encoded in neural responses. We know that human vision is mediated by a complex interconnected network of multiple areas that jointly together represent visual information. And each of these areas informs idiosyncratic representation of visual phenomena. They respond differently to visual stimuli. So over the last century, neuroscientists have tried to understand what is the representational content of each of these areas. And for example, seminal work in this direction successfully characterized early visual cortex for processing of oriented edges, or higher level cortical areas for processing of more complex stimuli, such as faces. But despite these advantages, we still do not understand fully the visual content, the representational content of visual representations. So here I will describe two steps that we took in order to further our knowledge into this direction. Now, the first step addresses the fact that different areas, say V1 and FFA, are typically studied in isolation of each other. But studying areas in isolation comes at the cost of an incomplete characterization of the visual system as an interconnected network. So when it comes to investigating representational content, we shifted our focus from asking what does each area represent to asking what are the representational relationships between areas. In other words, what representational content is shared between areas? What are the visual properties that areas are representing in a similar way? Or what representational content is unique between specific areas? What visual information is uniquely represented, encoded in specific areas with respect to other areas? So this was the first step, from studying areas in isolation to studying them as a network. And the second step that we took addresses the fact that representational content is often studied with limited experimenter-picked stimuli, but using small stimuli sets might risk to reproduce experimenter biases, while at the same time ignoring some important neural signals that would only be available from a broader sampling. So when it comes to investigating representational content, we used a large sample size to evaluate a broader, more diverse, and thus less biased hypothesis space. And to study representational content while following both of these steps, we developed a neural control method called relational neural control, RNC in short. So in order to address the first step, to investigate representational relationship between areas, the way RNC does this is that it finds across the entire hypothesis space of many images, it looks for controlling images that either align the responses of some areas or disentangle the responses, under the assumption that alignment will highlight shared representational content and disentanglement will highlight unique representational content. So aligning images with the images that two areas represent similarly, and disentangling images with the images that two areas represent differently. So then the goal of the algorithm is to find these images. And addressing the second step is to look for these controlling images while exploring a large hypothesis space. In order to do so, we apply RNC, so RNC search, on in silico fMRI responses to all 73,000 NSD images generated using the neural encoding dataset. So all the results I will be showing you, the results which are coming from in silico neural responses. And we applied RNC. We used RNC to investigate the representational relationship for human early and mid-level visual areas, V1 to V4.
So can I ask you a question?
Yeah.
Why don't you use the human data of NSD?
Okay, 3 reasons. So the first reason is that in the real data, single subjects only have up to 10,000 categories, whereas here I generated neural responses for all 73,000 categories for each subject. So there's a 7-fold increase in data. Second, I base my results on cross-subject validation. And if I used NSD, I could only cross-validate across 1,000 images, because those are the shared images, whereas here I can cross-validate across the entire solution space. Third reason is that encoding models learn to model the noise and the signal and not of the noise, except maybe for overfitting, but that is reduced. And because of this, the resulting in silico responses are denoised compared to real responses. And as a consequence, also the results of the algorithm are less biased by noise. it's a higher SNR.
So what's the content of the images in the different categories? Is it images of people or?
I will show later. I will show some examples, but it all consists of naturalistic scenes. So like daily things that you would see mostly on a daily basis,
Okay.
like giraffes. Yeah. I will give some examples later.
It's interesting to see it as a denoising technique because you're like not explaining all the explainable variants.
Yes.
So you're also losing some signal, right?
You're losing signal, but you're losing a bunch of noise.
Mm.
And as a consequence, you have less signal than the original data, but the SNR is still higher.
Yeah, I have to think about it.
Yeah. So we use this method to investigate early and mid-level visual areas. And specifically, since we wanted to look at representation relationships, we investigated representation relationships between pairwise comparison of areas. So V1 versus V2, V1 versus V3, V1, V4, and so on. And we started by using RNC to uncover representational relationships, so shared and unique representational content, between using on the univariate fMRI responses. So now by univariate fMRI responses, I mean the voxel average response across all voxels within one specific area. So an fMRI area V1 is composed of many different voxels, which are analogous to pixels in images. And here by univariate response, I just mean the average response over all these voxels, which are composing one area. And so in order to achieve this, in order to look for representational relationships, RNC searched the entire hypothesis space of 73,000 images for images that would either align the univariate responses of two areas, therefore highlighting unique shared representational content, or disentangle them, so to highlight unique representational content. And here you have, as an example, here you have the univariate response space of two areas on the x-axis one area, y-axis one area. So the alignment consisted of images that would either drive the responses of both areas or suppress the responses of both areas. Whereas disentanglement consisted in images that drove the responses of one area while suppressing the other, or vice versa. And importantly, as I was mentioning, we cross-validated these results across subjects, meaning that we found the controlling images using the univariate responses of 7 out of 8 subjects, and then tested them on the left-out subject until every subject was left out once. And here are the results. I will start with V1 versus V4. On the x-axis, you have V1's univariate response. On the, this is pretty small. Yeah. Yeah, so on the x-axis, you have V1's univariate responses, and on the y-axis, you have V4 univariate responses. The black cloud of dots represents the univariate responses for all 73,000 images, so the full solution space. And the colored dots correspond to the univariate responses for the controlling conditions. So you see that, as expected, the controlling condition that was supposed to drive both areas, here shown with the green dots, is on the top right corner of the manifold. So as expected. And the response, the condition, the images that would suppress both areas, they fall on the bottom left corner. And also the responses for disentangling conditions. So responses driving V1 while suppressing V4, the yellow, are on the bottom right corner. And as expected, and also expected, are the images driving V4 while suppressing V1. And another thing you can notice is that the manifold is quite correlated, meaning that the univariate responses for both V1 and V4 are quite similar to each other, actually with a correlation of 0.65. And here we have the same results, only plotted against the baseline. Here the baseline consisted, represented by those dashed lines, horizontal dashed line, and consisted of neural univariate responses for images that were randomly sampled from all the 73,000 images. And likewise, we also observe similar results for all other pairwise areas combinations. So together, this shows that RNC successfully aligned and disentangled the univariate responses for pairwise of areas. So we next ask whether there was a relationship between the distance of two areas, so how distant two areas are from each other, and how well they can be aligned or disentangled. And for this, we used the stepwise area distance, where areas that were adjacent to each other, for example, V1, V2, or V2, V3, V3, V4 had a stepwise area distance of 1. Areas that had another area in between, for example, V1 to V3 or V2, V4 had a stepwise area distance of 2, and V1, V4 had a stepwise area distance of 3. So here we asked, how well could the controlling images drive or suppress the univariate responses away from baseline as a function of stepwise area distance? And for this, we plotted the univariate, the absolute distance from the controlling scores and the baseline scores as a function of stepwise area distance. And what we observe is that for the disentangling conditions, so the conditions where the responses of two areas are differentiated, so one is driven and the other is suppressed, here indicated by the yellow and red dots. For the disentanglement conditions, the further away two areas are, the easier it is to disentangle them, which suggests that the further away two areas are, the more different the representational content is. And this is also supported by another finding, that when plotting the correlation scores from the image manifolds as a function, also as a function of cortical distance, we observe that this correlation decreases with increasing cortical distance, meaning that the further away two areas are, the less correlated they are, the more different they are. So together, this indicates that while a large portion of representational content is shared between areas, unique representational content increases as a function of cortical distance. So we then asked what the controlling images look like. So here I will show the images. And we did so. I will show you the results, the images for the V1 versus V4 comparison. So these are the images that drove both areas, that drove the responses of both areas, and they consist of they're pretty cluttered images with high spatial frequency content and multiple objects. And these are the responses, unsurprisingly, suppressing both areas. And they consist of empty scenes, mostly skies. These are results that drive V1 while suppress V4. They consist of high spatial frequencies, vegetation mostly. And these are the images driving V4 while suppressing V1, which tend to consist on one or multiple objects on low spatial frequency background, for example, an empty sky. And as expected, when plotting the baseline images did not show any consistent visual pattern, as they were randomly chosen. So from the disentangling conditions, or better, the controlling images from the disentangling conditions suggest that V1 is uniquely tuned and driven to high spatial frequencies, whereas V4 is uniquely tuned or driven to object-like shapes, which are also the features that are jointly co-occurring in the images driving both areas. So these images consist of both high spatial frequency or cluttered content, but also objects, and they both are absent from in images suppressing both areas. So together, this suggests that high spatial frequencies and object-like shapes are the visual features that lead to unique representational content for the V1 versus V4 comparison. So far, I showed you how we used RNC to investigate representational relationships, so unique shared representational content for univariate responses, so for voxel average responses. However, by averaging across the voxel, we're losing the potential signal that is contained in the voxel population pattern, meaning in the multivariate responses. And this is important because these multivariate responses, this voxel pattern, might contain complementary information, which is then destroyed when averaging across all voxels. So then, for the next analysis, we ask what are the representational relationships when taking into account the multivariate responses between areas. Now, a difficulty arises because multivariate responses of different areas are not straightforward to compare for two reasons. The first reason is that different areas typically have different amounts of voxels, meaning that they are of different dimensionality. And as a consequence, you do not have an easy mapping between the two. And even if they were of the same dimensionality, there would not be a one-to-one correspondence between voxel in area 1 and voxel in area 2. So in order to compare the multivariate responses, we need to find a way to bring these responses to a shared common format. And we do this using representational similarity analysis. So here I will really briefly talk about how representational similarity analysis works. So let's say that we have two stimuli, hand and umbrella. And these two stimuli lead to two different multivariate responses. So here in red is a cluster of voxels of one area. And you see that the activity of both cluster is different because brain responds different to different stimuli. So what we do then is we compare the multivariate responses for these two objects. We look at how dissimilar they are or similar they are. We correlate them with each other. And this gives an index of their similarity, which we then insert in a so-called representational dissimilarity matrix. So this is a matrix which indicates for each pairwise comparison of conditions how similar or dissimilar the corresponding multivariate neural responses are. And we can do this for V1, and we can also do this then for other regions, for example, V2. And the important thing to realize here is that by doing this, we are effectively transforming the multivariate neural responses from voxel space, which is not comparable, to image similarity space, these matrices, which are comparable. As you can see from the picture, they both represent the same images, and they are of same dimensionality. So then we compare the two different areas, say V1 and V2, by just correlating these matrices with each other. So then when it comes to multivariate RNC, we investigated the representational relationships with respect to visual information contained in the in silico multivariate fMRI responses of area V1 to V4. And again, RNC worked by searching across the 73,000 images for images that would either align the multivariate responses, thus highlighting shared representational content, or disentangle them, thus highlighting unique representational content. Where alignment consisted in images leading to similar representational similarity matrices, and disentanglement consisted of images leading to different representational similarity matrices. And again, we cross-validated the results across subjects. And these are the results that we got. Once again, RNC was successful in finding images that would either align the multivariate responses for two areas. Here, this is indicated by high correlation scores between the RSMs of two areas as purple dots. So here on the X, you have the different pairwise comparison. This is the correlation between the RSMs of two areas. And it was also successful in finding images that disentangled the two areas. Here are the orange dots. And once again, we asked whether there was a relationship between these results, how well areas could be aligned or disentangled, and how distant two areas would be for each other. So we plotted these results as a function of cortical distance. And also, again, here we found that for the baseline and disentangling conditions, there was a decrease in correlation as the distance between two areas increases, indicating that the more distant two areas are from each other, the easier it is to disentangle their multivariate responses. And once again, here we looked at, we checked how the images look like. So here, again, for the V1 versus V4 comparison, and these are the images aligning two areas. These are the images disentangling them, and these are the images, these are the baseline images. And the situation is not as straightforward as in the other case, but what we can notice is that the aligning images often contain uniform portions, for example, the sky on the upper half. The disentangling images did not, and the baseline images did so, but to a lower extent. So in order to gain further intuition, we looked at how the RSMs actually look like. These are the RSMs for the aligning images. As expected, they look really similar between the V1 and V4 areas. And you can also notice that their entries consist of really either highly positive or highly negative correlation scores. And whereas these are the images from the disentangling conditions, as expected, they don't look alike. Now, if we zoom in into the alignment condition, what we can see is that, let's take into account an image with a sky, for example. when we correlate the multivariate response for an image containing the sky in the upper half with another image containing a sky in the upper half, this results in a really highly positive correlation. But when we correlate an image containing the sky with an image not containing the sky, this is resulting in a highly negative correlation. So our hypothesis was that V1 and V4 are aligned, or this effect is driven due to retinotopic properties of V1 and V4. Specifically, due to retinotopy, uniform empty regions, for example, the sky, will lead to suppressed responses for the voxels that are tuned to this part of the image, and whereas the lower part of the image will lead to driven responses. So for this image, half of the voxels will be mostly suppressed, whereas half of the voxels will be highly driven. And this explains the high correlation that we observe when this image is compared to the neural responses for a similar image with a sky. And also explain why we observe a negative correlation when this image is compared to this image. Because here all of the voxels are correlated, are highly driven. Here only half of them are driven. So together this indicates that V1 and V4 are aligned due to shared representational content. So up to now we showed that the RNC solutions generalize across the in silico neural responses of different subjects. But to fully validate these solutions, we also had to check whether they generalize to in vivo responses, meaning to real brains, because all of the results shown so far were on non-real brains, computer-generated responses. So we did this for the V1 versus V4 comparison, and specifically we took the controlling images. These are the controlling images for both the univariate and multivariate responses. And we presented them to new subjects, independent subjects, in a new fMRI experiment. And as expected, we found the same effect for both the univariate case and also the multivariate case. So indicating that the image is successfully generalized to in vivo responses. And this, first of all, validates the RNC, the method, as a method to investigate representational relationships between visual areas. And at the same time, confirms the representational relationships that we found with the V1 versus V4 comparison. So to summarize this part, we used RNC to uncover the representational relationships, so unique and shared representational content, between early and mid-level visual areas using a larger set of in silico fMRI responses that are available in vivo, which led to both quantitative and qualitative findings. Quantitative findings are that a large portion of representational content is shared between areas, but we also found that unique representational content increases as a function of stepwise area distance, which could be due to either a decrease in anatomical connectivity with distance, or also to gradual changes along visual hierarchy, such as increasing receptive field sizes. On the qualitative side, we found that spatial frequency and object-like shapes determine the unique representational content of univariate responses, and that topological image properties, such as uniform empty regions, for example, sky, determine shared representational content for multivariate responses. And the interesting thing is that the visual features leading to shared or unique representational content changed between univariate and multivariate responses. Again, univariate was spatial frequency and object-like shapes. Multivariate was image topological properties, which indicates that univariate and multivariate responses, they capture complementary visual information. They capture complementary representational content, and in turn suggests that promotes the integrated analysis of different levels of neural responses, univariate, multivariate, but also others that we did not address here, such as, for example, latent spaces or geometrical properties, or also oscillations if we're dealing with time data. And how much time is left? Do you know?
This doesn't try to show the 5 minutes.
Yeah, but I don't know how much.
Just watch.
Okay, it's really short. Okay.
Not so strict.
Yeah, it's towards the end. So this part was be really quick. I just want to quickly address two of the main limitations of this approach. the approach of experimentation on in silico neural responses. So in silico neural responses come from encoding models. And the problem with encoding models, so predictive models of the brain, is that they have a limited prediction accuracy, meaning that they do not completely capture or describe neural responses. There are some parts of the neural response they cannot account for. And also they cannot generalize they generalize poorly out of the distribution on which they are trained. If you train them on a specific type of images, they will not perform well in predicting neural responses for different types of images. But the good news is that in principle, both of these limitations are overcomable. For example, thanks to initiatives that push for the development of better encoding models, which are, two of these initiatives are, for example, the sensorium competition, where it's organized in order to, it's a challenge in which participants compete in creating better encoding models of mice visual responses to stimuli, or BrainScore, which is a platform where everyone can submit their model, and then a benchmark, a leaderboard is updated in real time with the score of how well their model performs in predicting brain activity. But here I will focus on the Algonauts Project Challenge, which is an initiative initiated by our group, and specifically consists in a platform that aims at bringing neuroscientists and computer scientists together in building the best encoding models of the brain. And so we are currently in the process of preparing the 2025 edition of the challenge in collaboration with CNeuromod, which is this large-scale fMRI data set I told you about earlier. And specifically, we will give to participants, fMRI responses to over 60 hours of viewings of friends. So it's really large data set. And we will ask them to use these responses to train their encoding models. And then they will submit them to us, and we will check how accurate these models are. And the participants who resulting in the highest prediction encoding models will win the challenge. So we hope that this large amount of data, again, more than 60 hours per subject, will result in encoding models with state-of-the-art prediction accuracy. And then, at the same time, these models will be tested on out-of-distribution stimuli, so stimuli which are different than friends. So this will also promote encoding models with better out-of-distribution generalization. So to conclude, my vision for the future of computational neuroscience is that larger neural data sets combined with challenges will promote development of better encoding models of neural responses, which will generate more accurate in silico neural responses, leading to an increase of experimentation on these in silico neural responses, ultimately resulting in faster scientific development. And I thank my collaborators and funding sources who are contributing to this future.
Coming to the second talk of today, also from the Neurodynamics of Visual Cognition lab, Jiawei Li, presenting how do we understand language across different people and modalities, being a postdoc in the lab. So,
yes. Thank you, team. Thank you for the invitation. I'm so honored to present my study here. Also, I guess some people have some question marks in their mind. Oh, you are doing some language study while you are in a visual lab. I guess this is what I'm trying to answer today. And my presentation will be relatively short and just give a short introduction of two projects I'm doing. So yeah. So why I'm in a visual lab? This is maybe the first question I would answer. Yes. So mostly when people think about language, you are listening to something like now. But actually language information are coming from different sources. We listen to something, we read books, and that actually is a visual stimulate. And we sometimes, the language input is not single, like now it's just me talking, so you are just here like simple people talking. So this is like single input, but sometimes there was so many different people are talking like a cocktail party. So today I will just maybe try to cover these topics, how people like get information from different modalities, and sometimes how people pay attention to someone and ignore other information. So here comes our first topic. This one is called cocktail party effect, and this is like a work in my PhD work. So if you want to look, here's a paper, you can refer it. And so what is cocktail party actually effect? So cocktail party is some name or some like terminology raised by Cherry in 1955, and the thing is very simple. In a cocktail party, there were so many people are talking, but you can easily to follow someone and to ignore others. Maybe in Berlin they will call it like techno party effect, but so but you see, like the terminology was raised like 70 years ago. Why we still like talking about this one? So actually there was some little improvement of every region, every decade, I will say. So in 1953, they will only do like some behavior experiment, and in 1973, it's a science paper actually. They were trying to record EEG of a listener to listen to different things, but in the behavior experiment, they can like listen to a whole story. But in an EEG experiment, they only present the listener with like di 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 da, those like simple tone, because it's easy to analyze if you are familiar with ERP studies and things like that. And now in 2023, finally we can recording the EEG and like to like present the natural stimulate to the listener because of the encoding model, as Ali said, we were now finally can compute those things. So what is the advantage when you like ask the listener to pay attention to a whole story instead of like a very simple term? Because we can have more features in the speech. So when you listen to a story, you will hear the sound, and this is we call like acoustic information. Also, you can know the content of the sound. If you tell a story like a rabbit, an eagle is chasing a rabbit, and there is a river near the bridge, maybe you have some images in your mind. But if you present only di 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 da, something like very simple, you cannot get those kind of things. So there's like several things happening when you listen to a story. So we call them, when you hear the sound itself, it's the acoustic feature. And when you can understand a story, if I say some Chinese, 今天很好, I maybe in this room only few people can get their information, but this is the semantic information, not like the acoustic. If you hear some, you cannot understand that. And if you can, sometimes when you like listen to a story, there is a little pictures or little images in your head. Maybe this is something we are interesting, but not easy to give a name of it. So we see like in the like naturalistic speech, you can have like several different features. We use like different methods to represent them. Actually, there's a lot of work behind that, but today I will just simply like present them. For example, like the acoustic feature, you can use the sound envelope to represent it. For the semantics, thanks to the language models, also like early ancestors of GPT actually, we're using LSTM model here to calculate every word, to turn a word to a vector. Then we can put a vector to a computational model. We cannot put a word itself to the model. And finally, because we want to calculate some images in your brain. So this is not easy, but when someone is telling a story and someone is listening a story, there must be something similar in their speaker's mind and like listener's mind. So we calculate some kind of like correlation between the speaker and the listener, and we use a method called interbrain recording. It's very simple, so you just simply record the brain of the speaker. Usually we only record the brain of the listener. So this is what we done to capture different features. And the experiment is actually also simple. So we ask the speaker, we use the EEG to record their to brain activity, and we ask two speakers to tell the story, and we both record their their audio and their EEG. Meanwhile, we also invite like 20 listeners into the lab and ask them to pay attention. We put like two different sound in different years to ask them to pay attention to left or pay attention to right. So it's simple. And we use the method, we use the encoding model. Actually, I'm curious how many people has ever heard about encoding model. Ali is talking about encoding model, but do you guys really know what the encoding model is? Yeah. Maybe you can learn from Angela this semester. And to simply talk about it, it's like you put some feature like the sound, the envelope, and maybe like their semantic matrix. It's a vector, and you use some function to predict what the brain activity is, and to how to evaluate the model is good or not. You calculate the correlation between the predictive EEG and the real EEG. So what Ali is talking about is mostly based on the predictive brain activity, right? Yes, yeah. So we were like calculated the brain activity from the model and from the real one. So we can get some encoding correlation from this model, and we if you remember, you are asking the listener to pay attention to one and ignore another. So we can get like actually two plots from or two like encoding R temporal plots. So we can calculate are they pay attention if there is like huge difference between the people they pay attention to and they ignore. There were we say, oh, there was a attentional effect, or if there were two output are very similar and there was no huge effect between the attend and unattend, there was just no attention effect. So this is what we were basically working on, and what we found is there was like a different like critical time range and the region for the different features. So for the acoustic feature, it's the sound itself. It happens after 200 milliseconds to 300 milliseconds after the speech onset. So also it's happening on the theta band. It's a frequency band you will know or you will learn later. And there was like the region is like this. And for the semantic feature, it's happened a little bit later. It's around 215 milliseconds to 600 milliseconds. Also, there were a different frequency band. It's on data, not on data. And we have also a later like cluster, and cluster based permutation is like just a statistical way to like describe the data. So not so important. And also very interestingly, we calculate the correlation between the speaker and the listener, and we found something very, very interesting, and I like it. We found something before the speech onset. So when you ask the listener to pay attention to a speaker, when there was when the speech didn't onset, they already have some correlation between the speaker and attended the listener and attended the speaker. But not happen when the listener and unattended speaker. There was like a difference. Also, this difference is happening before the speech onset, not like not like after the speech onset. So we will say like the listener is like kind of predictively pay attention to the speaker. So yeah, this is this part by the speaker listener correlation, something like that. So we maybe summarize it. We found like the attention effect for the acoustic feature on theta band around 200 milliseconds, and after that is the semantic feature. Also, we found like speaker listener correlated before the speech onset. This is like my first study, and this is like this is during my PhD work, and now I will go to my second project, and this is like kind of like my postdoc project here. So for this project, I'm interested like in a very simple question. So this is simple motivation. We get information from different modality. Sometimes you listen to a podcast, sometimes you're reading news, but if there is something similar, we still have our rabbit and go. But we were curious because the input modality is different. So what is happening when you really understand something? So what is happening on your brain and how this like similar or semantic representation emerge in your brain. So we're doing the study. We present the listener this time participator to different story. There were some in listening in auditory modality and some in like visual modality. And of course, we present them like for the listening is easy. We just ask them to listen to some podcast. But for the reading, we kind of manipulate it. We pump up their words according to their time. Like they appear in the podcast. So this is what we were doing for the experiment. And because the encoding model, we need more data for the listening. So we ask the participant to listen more stories. So this is what we were trying to do with the encoding model. We trained the model on one modality, maybe like visual or like auditory, and we test the model like another modality, maybe like we train the model on visual modality and test the model on auditory modality, or we like switch it. So also we were not testing on one point, we were testing like on different time points, maybe not so easy to see. Also this, we call it like time generalization analysis. This is what we are doing here. And I can show like the preliminary result. Actually very interesting one. So we found like we train the model on the like the when we train the model based on the reading data and test on the listening data, when we use the occipital result, yeah, is here. So these are the occipital, and test on the central electrodes, they will have like the diagonal is exact time, like test on one point and train on one point, test on the exact same point, and like this means like something is leading and this is something like leading. But we can show like for the diagonal this is positive, and also like if we do another way around. So if we train the data on the listening, the central part, and test on the occipital, yeah, the reading data, they were kind of like flipped with each other, and they will also have like a really positive diagonal. Also something interesting like a negative correlation happened. So what happened if the speech not on set but in the listening, but they can like negatively predict the data like on the reading on the occipitals. We don't know, but we're trying to work on that. If you have more insights about this part, I'm really looking forward to hear that. So this is kind of what I'm doing here, and I'm summarize a little bit about the two projects. For the first project, I finished that, and this is we're talking about like cocktail party effect. We are interested in what our brain happening when you attend to someone and ignore others, and we found we can like before the speech onset, we can like have a correlation to the speaker you attend to, and we also found some different things on the acoustic feature and the semantic feature. Also for the cross modalities in semantic representation, we found it is not like exactly on the same region. You have like those kind of prediction, but they were like on the different regions. They have this kind of similar representation of the semantic. And for the methods, actually, we put a lot of efforts, like how to use those language models are now more popular. We use large language models like GPT-2 to help us, maybe now it's not GPT-2, GPT-4, something like that, to help us to do the annotation of the word itself. Also, I'm also interested in those computational model. What I'm using is the encoding model like Ali did. So this is the method I'm using. So if you're interested in the topic or interested in methods, I'm looking forward to like discussing with you. Thank you for attention.